Look around you. Now, take a look at your own hands. You're completely surrounded by and covered in a world of life so small it's totally invisible. So today, we're going to dive into that hidden world, the world of microbes. For most of human history, the rules of life seemed pretty simple, right? If it was alive, it was a plant or an animal. That's it. But then the microscope came along, and it didn't just show us new creatures, it revealed a world that just refused to fit into our neat little boxes. And that threw, literally, the entire system of biology into chaos. So this was what you could call the original classification crisis. The first microscopes didn't just show us new life, they showed us a fundamental flaw in how we even thought about life. The question suddenly wasn't just, what is this thing, but where in the world does it even belong? And you can see why they were confused. The old rules were just so simple. Plants, right? They're green because they make their own food. We call that autotrophic. They don't really move, and they can just keep on growing. Animals, on the other hand, are heterotrophic, which is just a fancy way of saying they have to eat stuff to survive. They move around, and their bodies have a pretty fixed size. It seemed like it covered everything, until, well, it didn't. Yeah, this simple system, it broke down almost right away. Early scientists, they really tried to just shove these new discoveries into the old boxes. So, algae that could make its own food, okay, that's a plant. Tiny little protozoa swimming around, well, they're like tiny animals, right? But it was just a messy, messy fit. What about fungi? They don't photosynthesize. And a ton of bacteria can move around on their own. The boxes were broken. It just didn't work. So they needed a new box. And in 1894, a biologist named Ernst Haeckel came up with a really elegant solution, a third kingdom, which he called Protista. This basically became the new home for all the biological misfits, everything from algae and fungi to bacteria. It wasn't perfect, not by a long shot, but it was a huge step. It was an admission that life was way more complex than just plants and animals. All right, so solving the classification problem was one thing, but actually understanding these new life forms, that was a whole different ballgame. This required a brand new type of scientist, the microbe hunter. These were the pioneers who were going to map this invisible territory. And this journey of discovery, it took a long time. It all started way back in the 1600s with Anton von Leeuwenhoek. He was a Dutch merchant who scraped gunk off his own teeth, put it under his homemade microscope, and became the first human to ever see bacteria. He called them animalcules, little animals. Fast forward 2 centuries, and you get Louis Pasteur, who figures out that these microbes are behind everyday things like fermentation. Then, Robert Koch comes along and proves they cause disease. And finally, Dmitry Iwanovsky discovers something even smaller and weirder, a virus. You see, for centuries, people believed that life could just appear out of nowhere, like mold on bread or maggots on meat. This idea was called spontaneous generation, and Pasteur just demolished it. Through a series of brilliant experiments, he proved that life can only come from pre-existing life. His conclusion, in his own words, was that it was the dust and air which carried the germs that caused spoilage. This was an absolutely revolutionary idea. Okay, but how do you prove that one specific microbe causes one specific disease? That's the real challenge. And Robert Koch gave us the answer with his 4 postulates. It's like a masterclass in scientific logic. Here's how it works. First, you have to find the microbe in every single case of the disease, but it has to be missing from healthy people. Step 2, you have to isolate that microbe from the sick host and grow it all by itself in a pure culture. Step 3, you take that culture and introduce it to a new, healthy host, and it has to cause the exact same disease. And finally, step 4, you have to be able to recover that very same microbe from the new victim. See? It's a complete, undeniable chain of evidence. It's the bedrock of medical microbiology even today. Okay, so now that we know how microbes were discovered and proven to cause disease, let's zoom in. What do these little organisms actually look like? What are they even made of? When you first look at them, their diversity is just stunning. You've got these tiny spheres called cocci, and they can form these long chains like a string of pearls or bunch up in clusters like grapes. Then you have rod shapes, which we call bacilli, and then you get the really exotic ones like spirals or the little comma-shaped vibrio. This basic shape, their morphology, is often the very first clue a scientist uses to figure out what kind of bacterium they're looking at. And on the inside, a typical bacterium is just a marvel of efficiency. It's got this tough outer cell wall for protection, and then a plasma membrane that acts like a bouncer at a club, controlling what gets in and out. The cytoplasm is inside, but notice what's missing? There's no nucleus. Its genetic material, just a single circular chromosome, is all condensed and just kind of floats in an area we call the nucleoid. It's life stripped down to the absolute essentials. Now, that cell wall we just mentioned, it's incredibly important. There's a staining technique from the 19th century called the Gram stain, and it divides almost all bacteria into two major groups based on that wall's structure. So Gram-positive bacteria have a really thick, tough outer wall. Gram-negative bacteria, on the other hand, have a much thinner wall, but is protected by an extra outer membrane. And the simple difference? It's absolutely critical. It affects everything from how we identify them to which antibiotics are actually going to work against them. So what is that wall actually made of? The key ingredient is this unique substance called peptidoglycan. The best way to think of it is like biological chainmail. It's this strong but flexible mesh made of sugars and amino acids that wraps around the bacterium to give it its shape and, crucially, to protect it from bursting under pressure. And a lot of bacteria aren't just floating around, they're active swimmers, and they're propelled by these incredible little molecular motors called flagella. These are whip-like tails that spin just like propellers, and their arrangement can be totally different. Some have just a single tail for zipping around in a straight line, some have a whole tuft at one end, kind of like an outboard motor on a boat, and others are covered in them, which lets them do these complex, acrobatic tumbles through their environment. So, we've looked at their history, their intricate structures, but knowing all this brings up a really critical, practical question. If these tiny organisms are literally everywhere, how do scientists possibly work with them without contaminating everything or getting sick themselves? I mean, really think about it for a second. The air you're breathing is full of microbes. Your hands are covered in them. So how in the world can you grow a pure culture of one specific type of bacteria without all the others getting in and ruining your experiment? Well, the answer is a very strict set of procedures. The whole philosophy behind working safely is pretty simple, and it's two-sided. First, you have to protect yourself from the microbes you're studying, especially if it's something that can make you sick. But just as important, you have to protect your pure culture from the countless other microbes in the environment, the ones on your hands, in the air, everywhere. It is absolutely a two-way street of protection. And this is all done through a strict set of rules called aseptic technique. You wear a lab coat and it's fastened to act as a barrier. You use a flame on the mouth of a culture tube because the heat creates an updraft of sterile air that pushes contaminants away. You never put caps or stoppers on the workbench. That bench is like a highway for stray microbes. Live cultures are never ever poured down the sink. They have to be sterilized first. And you store petri dishes upside down. Why? So that condensation, which is a perfect breeding ground for invaders, doesn't drip down onto your precious culture. These aren't just suggestions, you know. They are the fundamental rules that make modern microbiology even possible. So from a classification crisis centuries ago to the strict rules of a modern lab, our understanding of this invisible world has come an incredibly long way. And I want to leave you with this thought. Right now, as you're sitting there, trillions of these organisms are living in you and on you. Seriously, they're digesting your last meal, they're training your immune system, they might even be influencing your mood. We spent centuries hunting them, and now we're starting to realize they are an absolutely essential part of us. So what other unseen roles are they playing in your life right at this very moment?